So, chapter 16 brings us even deeper into what DNA is, how it works. Now, the beginning of the chapter, again, is kind of a lot of history of how this was discovered, but I think we pretty much covered that with the gene. I want to point out a couple experiments, make sure they actually make sense to you because they really are pretty ingenious. But we'll move on from that fairly quickly. We'll get into the meat of the chapter, which has to do with how DNA replicates itself. And then there's just a couple odds and ends, neat things that we should know about DNA. So let's jump in here. All right, so again, we're going to get into the nitty-gritty of DNA, okay? Whenever it feels like too much, just take a deep breath, step back, look at the big picture, right? If you understand inheritance, if you understand, the big picture of how these traits are passed on, it's okay. But by understanding, right, by breaking this down to its smallest piece, by getting very reductionist, we've gotten a lot of power. We've learned more things about how DNA works, right? So it was 1953 when Watson and Crick introduced this idea that DNA is a double helix. And really, from that, we've gleaned a lot of things, right? And now, in 2020, right, in these years, we can say that the hereditary information that we were talking about with Mendel is encoded in the DNA, reproduced in all the cells of the body, right? Every cell in your body has the exact same DNA, and this DNA directs all sorts of things, right? Right? It's not just inheritance. The DNA actually directs the development of biochemical, anatomical, physiological, all these traits, even some behavioral traits in some cases. All right, so there they are with their model. Okay, so a couple experiments that I wanted to emphasize here. So Griffith, they mention in the gene, and they're talking about his discovery of this idea of transformation. So transformation is when bacteria just pick up DNA from the environment and incorporate it into themselves. And so this is actually something really important to microbiologists or to doctors who are trying to treat disease, is this knowledge, right? We think of bacteria, we say they do asexual reproduction, but then they'll pick up this random DNA from the environment, and if it's useful, they will use it. So I just wanted to make sure that you understood this experiment because it's pretty cool. So here what they're showing us, right, is this particular strain of bacteria. I believe it's a Staphylococcus aureus. And notice how this is the one that's pathogenic. This is the one that kills the mice. It has this structure, it's called a capsule. If you take micro, we'll talk about it. But that basically keeps like the mouse's immune system from killing the bacteria. So instead the mouse dies, right? We have another version, right, of the same bacteria. It's basically the same species, but it doesn't kill the mice. Notice it's lacking that capsule. Now, if you take the lethal bacteria, but you cook it, right, you heat it up, you can kill that bacteria, and then when you inject it in the mouse, it lives. But if you put the heat-killed bacteria, right, it's dead, not going to harm a mouse, in with this non-pathogenic strain, right, the one that also didn't kill the mouse. Look at this. Here's the one that keeps the mouse alive and the one that keeps the mouse alive. But you put those two together in a petri dish, the living bacteria picks up the DNA, right, from the lethal, the killer bacteria, and it starts expressing that trait, that capsule. This is crazy that bacteria are doing this, right? But this was, I thought, a super clever experiment and taught biologists a lot about how that there had to be this molecule, right, that was moving hereditary information around. The other one that I wanted to point out, I love all these people are doing stuff with radioactively labeling things. You can still do it. some researchers do still use radioactive labels, but it's actually getting much harder to get the proper permits to do this sort of thing. But again, pretty clever here. So we're working with bacteria, and we're working with a specific virus that infects bacteria. These are called bacteriophages, or sometimes for short we just call them a phage. And so basically, they just radioactively label the two parts that make up a virus. Here they put the protein in pink, and here they put the DNA in blue. And the question was, well, which part of the virus actually ends up inside of the bacterial cell? And sure enough, it was that radioactive DNA. And so this was one of those experiments that they mentioned in the movie, kind of helping to solidify our understanding that it's actually DNA and not protein that's the genetic material. And it sounds so simple to us now. We're like, oh, of course. But it really is. They were looking for the language of life. And we know that there's like 20 different amino acids that the body uses. And you think about our alphabet and you go, well, we have 26 letters. That sounds much more appropriate to base a language on than the 4 letters that we find in DNA. So again, it's one of these things where it's like science is trying to help us understand the natural world. So just because something makes sense to us doesn't make it true, right? You got to figure out how to set up an experiment that can actually test that hypothesis, right? So DNA is the genetic material. But then it really isn't until, right, we start figuring out the actual structure of DNA that this is going to make more sense. So again, I'm kind of still before Watson and Crick here. Chargaff's rules, these are going to be important. We'll talk about this some more, right? Chemists start breaking down the DNA, right, and realizing the 4 different nitrogenous bases, right? 4 possible, this whole thing together, a phosphate, a sugar, and a nitrogenous base is called a nucleotide. So these 4 really nucleotides that we have here, right? And Chargaff's rules basically tells us that between species, the number of T's might differ. The T's and the A's and the G's and the C's may differ, right? Humans, may have 30% T, and chickens could have 17% T, right? It varies between species. But what he also noticed is that in any species, A's equal T's and G's equal C. And we didn't know what that meant, right, until we start getting this real layout of what DNA looks like. There's the famous photo, the X-ray diffraction that gave Watson and Crick that kind of key piece of information that helps them figure out this double helix. And again, someone had asked me, how do you turn that into understanding that this is what DNA looks like? And all I can say is it's like, if you were a music aficionado, right, and I don't know, say you love reggae, right, and you walk into a friend's house and you hear a song on the radio and you know it's Bob Marley, right? I walk into the house, I have no idea, right, who that might be. And so someone actually studying this can take things like, oh, well, that picture says there was a full turn every, 3.4 nanometers, that sort of thing. So you shouldn't be able to tell from that photo that it's a double helix, right? But here it is. It starts to make sense. And this is where Chargaff's rules come into play, right? The thing that we found was that DNA, right, from this image, that basically it's the same width across. And so what made sense there was that if A's and T's go together, or at least the single ring, the single ring, like a T and a C, those are called pyrimidines. These double rings, like A and G, are purines. And so we basically always need a pyrimidine and a purine together. And then with Chargaff's rules, we realize, oh, A always goes with T and G always goes with C. Okay, so then what you'll see throughout the rest of the book is there's multiple different ways that this can be drawn out, from kind of a fairly detailed come from a fairly detailed look where they're actually showing you that base pairing, maybe an image where they're just showing the double helix, right? That backbone, that outer portion that's made by repeating sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, right? Here's a little unwinding the double helix, so kind of laying it out flat, maybe abbreviating. Right now we're not showing the sugar and the phosphate, just the base pairs, maybe just 2 pairs, maybe just 2 lines, right? So there's multiple different ways that you could lay this out. And there's our A and T, G and C. Now, the cool thing here is, do you remember a dotted line bond between a hydrogen and another atom? It's a hydrogen bond. So it actually turns out that it is hydrogen bonding that holds the 2 strands of DNA together. Was a hydrogen bond a weak bond or a strong bond? It's fairly weak, right? We can actually pull that apart. When you boil water, you're breaking those hydrogen bonds to release it to the atmosphere. So compared to a covalent or ionic bond, that hydrogen bond is fairly weak. And so that's going to mean that we can actually pull the DNA apart, right? And so this, again, Watson and Crick started figuring this out, this idea of replication, right? DNA, if it's going to be this genetic material, what's carrying all this heritable information, we have to have a way to copy it. Okay, so we're going to spend a good chunk of time here. This is the nitty-gritty of DNA, looking at how DNA is replicated. One thing that students often find super confusing, let me take a moment to talk about this. So they do point out, right, that DNA is what we call anti-parallel, right? So parallel means 2 things running in the same direction. So then you get a picture DNA being anti-parallel, right? They're still running alongside each other, but kind of in opposite directions, right? So if the 3' end is up here, then on the complementary strand, the 3' end is at the other end. And you're going like, who cares? And let me show you where this, where does this, see if I have a good picture. Okay, let me show you where this comes from, right? So notice again, on this slide, they're pointing out here's the 3' end, and here's the 3' end. Okay, so basically what happens is chemists number their carbons. So remember, so this is a deoxyribose. It's a 5-carbon sugar. And remember when we were talking about chemistry, we said this is shorthand. Anytime there's like a little intersection, oh, sorry, I'm circling it, but that doesn't show up, right? Anytime there's a little intersection, that's actually a carbon. So what chemists do is they number their carbons. So this is the 1st carbon. This little intersection here is the 2nd carbon. Sorry, that's terribly difficult to read. Here's your 3rd carbon. 4th carbon is at this intersection. And then that one right there is your 5th carbon. Anybody see it? The 3rd carbon is that 3' end. The 5th carbon is the 5' end. Okay, so that's where you get the numbers. It's just which way the sugar is facing. Notice this sugar is flipped over. I don't know if I can do this, but I'll try. So here's carbon 1, 2, 3. Oh yeah, I can totally do this. 4, and here's that 5. So, yep, there we go, right? So there's the 3' end and the 5' end. Okay, what you need to know is that basically like this 5' end, it has this, phosphorus, 5 phosphorus, right? It has this phosphorus on there. And the way that DNA is built, you can't actually add anything on that end. It's kind of like that end is blocked. So if you wanted to make this strand longer, you always have to add on this end. And so what that means is when you're building a strand of DNA, it always elongates, and this is true for RNA too. We'll get there. It always lengthens or elongates from the 5, and that's prime, 3 prime, from the 5 towards the 3. You can't add to 5, so you always have to go towards 3. Okay, so in this picture, that would mean if we were building this DNA, this strand could grow this way, and this strand could grow that way, right? That's an old arrow. Okay, so they're anti-parallel, and that's going to be really important in replication. So that's why I wanted to come back here. Okay, so Watson and Crick are looking at, what is the possibility? How does this work? And when they envisioned it, it went like this, right? You have this parental DNA strand, right? Just if before a cell is going to divide, we're calling it the parent, right? And they're picturing that the DNA would separate, and then each side would get a new complementary strand. Now, they called this a semi-conservative model, meaning that each strand ends up with one half of it being parental, see how it's still in dark blue, and then half of it being that new complementary strand. So they called it semi-conservative. Only half of that was the old parental molecule. But there were other hypotheses of how this might work, right? That sure, DNA acted as a template, but here's our whole new model, right? And here's our parental strand. This is what Watson and Crick's would look like, right? Each time it gets copied, you get half the parental strand and half new DNA. And then this idea of the dispersive model is kind of like pieces of DNA going everywhere. And so again, it's like, hey, bust out the radioactive substances. So this time we're actually growing bacteria in a radioactive nitrogen. And so they use 2 different isotopes so they could differentiate between the first replication and second replication. Anyway, kind of cool, right? We could actually see that after the first reproduction that there was a mix of new and old. And so already we could kind of get rid of the one hypothesis that there was this conservative model. But we actually went to a second replication, right? And that's where they're using a different isotope. And then we can see, well, no, it's not evenly like distributed new and old nitrogen, right? We can see that they're parental strands and these complementary strands. So anyway, DNA. And now when you think about it, this makes perfect sense, right? It pulls apart and each side acts as a template, adding new information opposite it. Okay, so this is now, we are getting into the nitty-gritty DNA replication. So what we're going to see is that to make a copy of DNA, it's always going to start at what we call an origin of replication. This is a place where the 2 DNA strands are separated. They open up into this little, what we'd call like a replication bubble. So over here, they're showing us what this looks like in E. coli, right, in a bacterial cell. So bacteria have a single circular chromosome. And so when they do this, they would open up an origin of replication, right? And the DNA would start copying and almost just like flip open, right? It's totally getting copied, one parental strand in dark blue and then one kind of daughter strand there in light blue. In eukaryotic cells like our own, this gets a little bit trickier, right? So we have linear chromosomes. We've been looking at those. And so what ends up happening is you get this same little replication bubble, but we actually might end up opening up multiple spots along the DNA for a series of enzymes to come in and start working at the same time. But basically, in both groups of organisms, replication is going to start proceeding in both directions from that origin of replication until the entire molecule is copied. Okay. I think they did a really nice job with this artwork. So there are a lot of enzymes involved in this replication process. So an enzyme is a protein that catalyzes a reaction. It speeds up a reaction. So it's not like forcing things to happen, but it's encouraging them. And we'll look at that. Here's a hint for you, particularly if you're going on in biology or healthcare. when you see a word that ends in ASE, that's a really good indication that you're dealing with an enzyme. Okay, so let's look at a couple at what's going on here. So we're kind of zoomed in now on that replication bubble. And at each end of that bubble, we're going to have what's called a replication fork, right? So it's kind of shaped like a Y, and this is where the new DNA strands are going to elongate. So our first enzyme that we're looking at here is an enzyme called helicase. It actually untwists the double helix, it separates, right? It unzips the DNA. And then they're showing, these in gray here, these are what are called single strand binding proteins. So the double helix, when it's doubled, is really stable, right? It's got all those hydrogen bonds. Everybody thinks they have enough electrons. Everyone's happy. And so these proteins are going to stabilize the DNA while it's in this single stranded form. If you picture helicase kind of moving along the DNA and kind of unwinding it and unzipping it, one of the things that's going to happen is kind of over, in this direction, right, in front of where the helicase is going, picture, I don't know, what's a good example? So if you like twisted a string and you say stuck your finger in the twist and started pulling along it, right, unzipping it kind of like helicase would do, in front of your finger, the string would get super coiled. That's exactly what's happening in the DNA, right? It's getting like super bunched up. And so this is another enzyme. It's called topoisomerase, and it kind of goes in front of the helicase and cuts, like snips the DNA so it can unwind, get rid of all that extra tension, and then allowing it to kind of swivel and rejoin itself. Okay. From there, so now we have this open replication fork, and to kind of start things off, what we're going to use to mark the area where new DNA should get built is a little segment of RNA. So now they're putting this in red. It's called an RNA primer. It's just a short segment of RNA. It's something like 10 nucleotides long that is going to indicate this is where our next enzyme, DNA polymerase, should start. Okay, so lots of enzymes: helicase, topoisomerase. There's an enzyme primase that actually inserts that RNA primer, right? It helps kind of make sure the right RNA nucleotides are finding their way. Okay. Again, here's what we were talking about earlier, this idea that you can only, here's our additional, our incoming nucleotide, right? Notice if we had an A on the template, that T wants to be there, right? It's actually attracted. They fit nicely together. We're going to get a hydrogen bond going here. But notice we're only adding to this 3' end. And so this is where this reaction takes place and adds to our strand. Okay. So okay, this gets a little tricky. So basically what happens, because the DNA is anti-parallel, it can't be built the same on both sides, right? We can only add to the 3' end. We can only build 5' to 3'. Here's the other thing that gets students. When I say 5' to 3', I mean on the new DNA strand. So like, for example, if you look here where that RNA primer was, it is at the 5' end of the new strand, right? And it's going to build this way towards the 3' end. For the template, it would look exactly opposite, and students always get confused. They're like, wait, isn't that going, the wrong way? But it's of the new strand. Okay, so what happens here is the DNA polymerase is going to be, and this is DNA polymerase 3, actually, starts adding new nucleotides, the complementary bases, but only on the 3' end. And so on one strand, that can happen continuously, right? As helicase is unzipping the DNA in this direction, this DNA polymerase is just DNA polymerase 3, is just going to keep going in the 3' direction. And so we would call that the leading strand. The leading strand is able to be synthesized continuously, here you go, in the 5' to 3' direction. we're going to run into some trouble on the other end. So anyway, I like this little zoom in here, right? If this was our origin of replication, we inserted a primer and off we go as the leading strand. Okay. Now at that same replication fork, the other side is going to be called the lagging strand. It can't work the same way. In fact, we'll see it gets built in these little chunks. And so let's look at that. Okay. Sorry, this is just zoomed in of what we were just looking at, right? That continuous building. Here's our lagging strand. It looks complicated, but notice what happens. So we're zooming in to our origin of replication again, right? So right where the DNA gets opened up by helicase, we put a primer in. There's primase, adding that RNA primer. But now we have to build, again, 5' to 3'. So we're actually going to build back towards the origin of replication, right? And as we get that done, then primase is going to move back this way and add another RNA primer and let us build back towards the first. So the lagging strand is actually getting built in little chunks. They are called Okazaki fragments, right? So there's our Okazaki fragment number 1. As soon as that, and this is kind of happening simultaneously, right? Helicase kept going, kept unwinding, and so primase is going to put in another primer, and now we're building our second Okazaki fragment. So it's still DNA polymerase 3 that's helping to build the Okazaki fragments. But then what we've run into is we have a lot of primers left in that lagging strand. And so here they're showing you another enzyme called DNA polymerase 1. DNA polymerase 1 actually comes in, pops out the RNA, and puts the correct DNA in, right? So we see it removing the red RNA, and it's going to leave all blue. There's also then just like a little gap, and so to hook neighboring Okazaki fragments together, we have one more enzyme. It is called ligase. I'm attempting to underline that. There we go, ligase, and that fuses those. I'm in your way, aren't I? Let me get out of your way. Right, so here's ligase, and it's just making sure there's no gaps between Okazaki fragments. Okay, there's better recordings out there. Let's see, I probably actually can't even show you this. In class, I'll pull up a good animation, but do realize if you go to your e-text for a lot of these things, they've put in little animations. The voices are super, I don't know, boring, I think. But sometimes you need that visualization, right? This is also, it's so tiny. How do we even know this is happening? It's pretty amazing. So this is a nice table in your book, so table 16.1, and it goes through those different enzymes and what their role is in DNA replication. You want to know what these are, what they do, what order they do it in. Heck, I would like print this out and just like cut out little strips, fold them in half, make flashcards, right? They'd be totally ready for you. But again, briefly, helicase unzips the DNA. That single-stranded binding protein just stabilizes that single-stranded DNA. Topoisomerase hops in front of helicase to get rid of any extra supercoiling that's taken place. Primase is who's adding the RNA primers. DNA polymerase 3 finds that primer and then starts adding DNA, again in a 5' to 3' direction, always. After that, DNA polymerase 1 comes in and removes the RNA primers and adds DNA. And then finally, the DNA ligase fuses adjacent Okazaki fragments. Okay, so think through that process a couple times, maybe look again for some video animations. I'll share one in class that I know is pretty good. Okay, so that is DNA replication. Now, well, all of that sounds crazy, right? You've got all these enzymes, all these tasks. This is happening in the nuclei of cells basically if they think they're going to divide, right? You have to copy your DNA before you're able to undergo mitosis or meiosis, as we were talking about. And so it's not like every cell is doing this all the time, but cells that replace themselves pretty frequently, say skin cells, this is happening really frequently, and it happens really quickly too. These enzymes are just flying across the DNA. And if you think about it, you've got billions of letters that need to get copied, and you don't want any mistakes, right? A mistake in the DNA code is a mutation, right? And we know mutations leading to cancer, mutations can lead to death. Generally, you got a good thing going, you don't want to screw it up, right? You don't want a mutation. So now we're into that, like we're done DNA replication. Now we're into some of those final kind of cool things that you can do with DNA. I thought this is really neat. So you don't want any mistakes. And so basically, we're going to do some proofreading here, right? And so this is an example. This is what's called nucleotide excision repair, right? That's a really fancy way to say, oh, we put the wrong letter in. We're going to take it out and fix it. And so you have another enzyme, this big green blob here called nuclease, right? I can tell it's an enzyme, ACE. It cuts it out and replaces that damaged piece, right, of DNA. So we bring that DNA polymerase back, right? We cut out the damage with the nuclease and say, get back over here and do it right. And now the DNA polymerase is going to make sure that the correct base pairing takes place. So it's kind of neat. DNA polymerase is really good at its job. Like for how many letters it has to copy, it makes very few mistakes, but then we proofread it, just like you should proofread your papers. Okay. Ligase comes in and makes that final connection. Okay. So when I talk about mutations, mutations generally are bad. I should point out, though, that mutations are also the source of genetic variation that natural selection operates on, right? So if, if you have a moth, if you never have a mutation that makes some of them, dark colored, this is like the classic example. You have like moths on a tree during the Industrial Revolution, and maybe like white moths were more common before, but now that the trees are covered in soot in the Industrial Revolution, the dark ones are camouflaged, right? So the birds don't kill them, they survive and reproduce. But if you never had that mutation that made one moth darker than the other in the first place, you wouldn't have anything for natural selection to work upon. So sometimes those mutations get through the proofreading, right? And they are actually really important for natural selection. Okay, other things. So this is pretty wild. So bacteria don't have this problem. Eukaryotes have a problem because we have linear chromosomes, right? And so they have an end, where bacteria, since it's circular, there's no beginning, no end, it just gets copied. Boom, we got 2 copies. But what we're going to see happen here is that basically we run into a situation where DNA polymerase can only do so much. So it basically sets up this problem where this one end of the DNA, where we placed an RNA primer, since you can't add to that 5' end, there's no way to finish this molecule, right? So the DNA polymerase has no way to complete that 5' end. And so each time you replicate the DNA, that DNA strand actually gets shorter and shorter, right? So that's kind of what they're walking you through here. So like, okay, well, we lost that, not the end of the world, but the next replication, right, we lose a little bit more and a little bit more. So what eukaryotes have done, which is pretty ingenious, is at the end of each chromosome, they have a special region of DNA that are called telomeres. So telomeres are basically some of that junk DNA that they talked about in the gene. They don't have any genes coded for them. And so they provide kind of this extra DNA so that it doesn't matter if the DNA gets a little shorter and a little shorter and a little shorter, but only to a limit. right? So what we'll find is that most eukaryotic cells are actually limited to about 50 replications before they run out of telomeres, and that cell won't divide anymore, right? It can't copy all the DNA it needs. So this has been a really interesting point of research in some of the things with aging, right? Considering, is telomere shortening part of this aging process? There is an enzyme called telomerase, there's your ASE ending, that actually lengthens telomeres. And we'll find this in humans, we find this in the germ cells, right? So in eggs and sperm, to make sure that each replication, how do I say this? So that you're not shortening with each generation of the human population. You actually have an enzyme to kind of rebuild those telomeres. We've also found this enzyme in certain kinds of cancer. So cancer doesn't just replicate 50 times and then stop. It can go out of control over and over and over again. In fact, one of the really famous, sounds weird, famous cancer lines, we have breast cancer cells from 1951 that are still dividing. So what are we, like 70 years later? That is not normal. That cancer is expressing this enzyme telomerase, and it just keeps making sure it has enough ends on either end of its important DNA that it can keep replicating. So that's kind of an interesting, right? Again, like if you learn, how do I say this? This is the difference between basic and applied science. So learning about telomeres was super important, right? Like understanding this nitty gritty of like, each time replication happens, we're losing a little bit. And then that leads us to this understanding of telomeres. And again, a lot of people are like, well, who cares? Why are we messing with this? But then you realize it's important to cancer, and maybe that would actually be a way we could treat it, right? If we could stop the telomeres from lengthening, maybe we could cut off the life of this cancer. Anyway, basic science versus applied science, actually coming up with a treatment or an improvement because of that. Okay, another thing I wanted to point out deals with how DNA is packed inside of the nucleus, right? Because it's pretty wild. You actually end up with a few meters of DNA in each cell, right? And we looked at some cells in lab, they're tiny. And so somehow packed inside the nucleus, we have to fit all of this DNA. And so that's what we're looking at here, right? Is that our double helix is basically it's wound around these proteins called histones. And then you see how it's looped and looped and looped, right? And we managed to cram this all into a chromosome. So a chromosome is made of the molecule chromatin, which is the DNA, and the protein that's called a histone. That's chromatin. So then that's wrapped up into the chromosome. Now, what I wanted to point out with this is that this actually ends up being important in how we use DNA, right? So if I had a place where, say, that was nice and like loosely packed, right? There's space between the histones. There's kind of some exposed DNA there. Loosely packed chromatin is called euchromatin, EU chromatin. And basically, when we are going to, how do I say this? Tightly packed chromatin, right? Like what we see over here, super kind of coiled up, is called heterochromatin. So what we'll find is that when the DNA is packed as heterochromatin, it's really difficult to actually use any of the genes. It's like they're packed away. Remember our bar body, right? That second X chromosome, that's going to be shoved together as heterochromatin. You're never going to use that DNA. You need euchromatin, you need loosely packed chromatin in order to actually access the information in the genes. And we'll talk about that more with some gene expression. And I just thought this was super cool. I hadn't seen this before. Your book is talking about, this is showing an image of the nucleus, and they've actually kind of tagged different chromosomes in different colors and noticed where they hang out in the nucleus. I thought that was pretty neat. Okay, so a lot going on there in chapter 16. Bring your questions on Monday. Let's talk about it more. Let's make sure it makes sense because we're going to kind of keep building on this, right? Next, we're heading towards, okay, that's cool. We got DNA. How are we actually going to express it? Don't forget to write about the gene, and I look forward to talking about that with you guys some more as well. All right, bye.